Before we can learn about epistasis, we must introduce two concepts, the first being genotype. A genotype is the set of genes a person inherits from their parents. In genetics, alleles are letters used to represent genes so that inheritance may be predicted. The next concept is phenotype. This is the physical manifestation of a genotype. For example, this horse's genotype contains two dominant A alleles and two dominant B alleles. Together they encode for dark brown pigment, which manifests in the phenotype as a dark brown horse. Epistasis occurs when two organisms have different genotypes but the same phenotype. These two horses have different genotypes for coat color, yet both are chestnut. Something must be occurring at the protein level below what we can see to cause this discrepancy. Epistasis is best visualized using pathways. Here we have a pathway for horse coat color. This trait is mediated by the interactions between two genes, A and B. We will start with a chestnut horse. If this horse inherited two little a alleles from its parents, it would fail to make enzyme A, which would result in a chestnut-colored horse. On the other hand, if it inherited one or more big A's from its parents, it would produce enzyme A. However, a horse that only makes enzyme A would still be a chestnut horse, because in this case enzyme A only produces the precursor necessary for enzyme B. Enzyme B is only present if the horse possesses a big B allele. Only a horse that produces both enzyme A and enzyme B would present as a dark brown horse. A horse with two little a alleles would always be chestnut color, since without enzyme A, enzyme B does not function. There are many manifestations of epistasis, but this kind is called duplicate recessive epistasis. Let us predict the genotypes of the cross between two dark brown horses that are heterozygous for both A and B genes, meaning that they have one big and one little allele at each gene. First we combine the mother's A genotype with the father's, in the Punnett square. Then we do the same with the B gene. Now we construct a branch diagram to determine the likelihood of any given offspring genotype. The big A allele appears in 3/4 of the offspring, while homozygous little a only appears 1 out of 4 times. Independent of the first gene, any of the offspring have a 3/4 chance of also possessing a big B allele and a 1 out of 4 chance of inheriting two little b's. Then we simply multiply our fractions across and combine the genes. We can take our probabilities and compare them to our epistasis pathway to determine the offspring phenotype probabilities. Any offspring with two little a alleles will be chestnut since they do not possess any of the enzymes. This makes 4 out of 16. Additionally, any offspring that can produce enzyme A but not enzyme B will be chestnut since they only synthesize the precursor to the pigment. Lastly, any offspring with a big A and a big B allele will be dark brown horses since they produce the precursor and they have the enzyme to synthesize the pigment. Now we can do the same thing but with different parents. This time it is simpler. We can compress the Punnett squares since some genes have duplicate alleles. This time half of the offspring will make enzyme A and half will not. The B gene is even more simple since all offspring of this cross will make enzyme B. Then we just multiply across to reach our final genotypic probabilities. Now compare our genotypes to the epistasis pathway. All of the offspring will be able to synthesize enzyme B, but half will not be able to make enzyme A, producing a chestnut horse. The other half will be able to, resulting in a dark brown horse. This produces a phenotypic ratio of 1 chestnut to 1 dark brown.